Hello, everyone. Welcome. My name is Jennifer Novotny, and I'm the Public Programs Coordinator here at the MIT Museum. Thank you so much for joining us for our first of 3 Soapbox events. As you can see, we have 3 wonderful evenings planned for you. Tonight, we're focusing on transmission. October 5th, we're talking about communication. And October 12th, we're talking about treatment. So I hope you guys will consider coming and joining us for the next 3 Wednesdays. Our Soapbox event might be a little bit different than most science talks you've been to in the past. So the way the format of this evening is going to go, our 3 speakers will each be giving short presentations about their research, and then you guys will have an opportunity to discuss in your own groups, tables. So you guys can talk about what you've heard and have an honest conversation about your thoughts, things you wonder, and things you'd want to keep knowing. We're going to pass out iPads, so each table can also tweet in questions you have, and we'll have a live stream of the questions. So that might inspire a conversation at your table, or it might be an inspiration for, at the very end of the evening, we'll have an opportunity for a Q&A session with our panelists as well. At the end of the evening, we will be handing out a very short survey. It should take you about 3 minutes. And if you fill it out, we'll enter you into a raffle for a pair of passes to come back to the museum and some really cute plush common cold viruses. So hopefully you'll take the time to do the survey as well. So with that, let's get started with our first speaker, Angela Early. Angela is a postdoctoral fellow at the Broad Institute, where she's working in the Genomics Center for Infectious Diseases.
Okay. Hello, everyone. Thank you, Jennifer, for putting on this great event, and thank all of you for being here today. I'm really excited to have this chance to talk to you a little bit about some questions that are near and dear to my heart, and have the chance to talk to Caroline and Erin about their research as well. As Jennifer mentioned, I'm a postdoc at the Broad studying malaria in the Center for Infectious Disease Dynamics. My background is in ecology and evolutionary biology, so in studying diseases, I tend to take this evolutionary genomics approach, trying to understand how diseases have evolved with their host, or in the cases of something like malaria, with the vectors that transmit them. This means that I spend a lot of my time thinking about this little critter up here, the mosquito. We just finished summer. We all have had, without a doubt, many sort of close encounters with what most of you probably consider a very annoying and obnoxious insect. Very few of us, however, would probably put it in the same category as all these other animals that I have pictured up here. And just think for a second, and if anyone wants to venture a guess, how would you place the number of deaths caused by the mosquito in relation to the number of deaths caused by one or a combination of these predators that I've pictured here? Any brave answers that want to get ventured? Yeah? 3 orders of magnitude. Well, it's in the right direction, but not quite in the right orders of magnitude. The mosquito is pictured down here in red, is estimated to be responsible for close to 750,000 deaths per year. And the only animal that even comes close to competing with those types of numbers are actually human beings ourselves. This has earned the mosquito the name of being the most deadly animal. But of course, it's not the mosquito itself that are causing these human deaths. What the mosquito is doing is transmitting diseases from infected people to healthy people. These are classes of diseases known as vector-borne diseases. And while mosquitoes are very common vectors, many other diseases that plague the world and that we're even cognizant of here in the northeast of the United States are also vector-borne. For instance, mosquitoes, you can't look at the news today almost without coming across mentionings of Zika. We've also had episodes of West Nile virus in this area of the country. Most of us are worried about Lyme disease here in New England. So mosquitoes, fleas, sandflies, ticks, even pictured on the right there, aquatic snails in some parts of the world are able of transmitting diseases. Of course, when you're having a vector-borne disease, then what you're going to be interested in is not only studying the biology of these actual diseases I have listed here at the bottom, but also understanding the biology of those vectors on the top, because it's only understanding both pieces of that puzzle that we can actually start to interrupt this transmission cycle. So as I mentioned at the beginning, the disease that I spend most of my time studying is malaria. It's transmitted by this mosquito vector on the left, known as Anopheles. Now, around the world, we have about 3,500 species of mosquitoes that we know about, but only about 30 to 40 of those are actually capable of transmitting malaria. And the main malaria vectors are all within the same family of mosquitoes called Anopheles. Then on the right, what I've actually pictured, what's stained there in purple, is the actual parasite that is responsible for the malarial disease that you would have in your body. And this is a parasite called Plasmodium. And just as there are numerous mosquito species that are basically benign, there's numerous Plasmodium species that have no way of infecting us. There's only about half a dozen Plasmodium species that can actually infect humans, whereas the other Plasmodium species we know about infect a range of hosts, including our cousins, the primates, but they can also infect birds and even reptiles like lizards. So in this broad sense, malaria falls into this pattern of a vector-borne disease. A mosquito will feed on a human host, and when it sucks up blood in that blood meal, it'll also take up malaria parasites that it can then transmit to an otherwise healthy individual. In order to really understand and hopefully eradicate malaria in our lifetime, however, we need to fill in more details in this cycle. And what I'm going to do in the next few minutes is just sort of give you an overview of the research that's going on in these areas and talk about attempts to sort of break this transmission cycle and hopefully bring about an end to malaria. So one thing that would probably automatically pop to everyone's mind is, well, you can simply treat the disease malaria in people. If you eliminate the malaria in our bodies more rapidly, then there's going to be fewer opportunities to transmit that malaria to a mosquito, thereby infecting another person. And this, of course, has been the approach that's been taken over centuries in malaria and millennia even, as even through traditional healing remedies like quinine, which are originally taken from the bark of a South American tree. So we have drug treatments. There's vaccinations that are not currently deployed but are in development. And we can also improve diagnostics so that malaria infections can be recognized and treated more rapidly. There's a liver stage, however, that occurs in the disease before the parasite moves into the blood. And when the parasite is here in the liver, the person's actually asymptomatic. You have no fever, no chills, no anemia, and so as far as the doctor is concerned, you're not yet sick. So a lot of efforts now are going into blocking this initial stage before a person has even manifested symptoms and before they can be transmitted to a next mosquito. And these types of liver stage vaccines are actually an area of the research I'm currently performing here at the Broad, trying to understand how the immune response acts towards malaria, and if we can understand that better, we can tweak vaccines to better boost our natural immune responses. Of course, this is ignoring what I said at the beginning about needing to take into account these vectors as being a really key part of this transmission cycle, and this is, in fact, a huge area of current research, both here in the Boston area and globally. In order to do this, then, we need to look not only at the mosquito itself that's causing these diseases, but you can put it in the context of all those other 3,000 mosquito species that we know do not transmit malaria. What is special about the actual malaria vectors that are allowing them to passage this malaria on? For instance, this mosquito I have here in the middle is the only one of these 5 that is actually capable of transmitting malaria to humans, but by looking at this, you would have no idea of knowing that. So one way that research is now helping us understand is through the development of transmission blocking vaccines. So these vaccines may seem a bit controversial because they would not actually help the symptoms in the patient themselves, but what these vaccines would do is it would alter the form of the alter the form of the parasite that is found in the blood, so that once that parasite is taken up by the mosquito, it is then destroyed in the mosquito and does not have a chance to enter its next reproductive cycle and be passaged on to another person. There's also ways, though, to simply block the mosquito from biting the human to begin with. Some of these may seem sort of obvious. You can put on insect repellent to prevent someone from biting you, but understanding the biology of the mosquito also means understanding why mosquitoes are attracted to humans to begin with. recent research has discovered what odorant receptors the mosquito uses to distinguish a human from a chimp from a cow. And this information can be used to develop better insect repellents. Understanding the ecology of the mosquitoes also feeds into intervention practices like bed nets. So bed nets have actually been responsible for a huge drop in malarial deaths over the last decade. But they only can be deployed in areas where malaria is mainly transmitted by nighttime biting insects. If malaria is mainly transmitted by daytime biting insects, having bed nets at night is not going to have a major impact in an area. And finally, one of the main areas of sort of current research is trying to simply reduce mosquito numbers. And this has come up a lot in the news regarding Zika outbreaks as well. Again, there's sort of time-tested approaches like insecticide spraying and habitat drainage. These two approaches are largely what eliminated malaria here in North America in the mid-20th century, largely through widespread use of DDT. But there's more modern and also genetic approaches that are currently being actively researched. This includes sterile male matings. Here what you can do is release large swarms of sterilized male mosquitoes. These male mosquitoes can mate with the female, trick that female basically into thinking she's capable of laying eggs. But then when she actually goes to lay eggs, those eggs are all sterile and no new mosquitoes emerge. Similarly, there's genetically modified mosquito approaches that are currently under research. This could include similar sterility approaches, although using genetic techniques rather than really labor-intensive irradiation. But other ways of releasing mosquitoes that could or sort of alter the dynamics in key areas are also being explored. So in conclusion, these vector-borne diseases, both malaria as well as a whole slew of other diseases, are all part of this complicated cycle that scientists are currently trying to understand. And by blocking any one of these, we can start to break down this cycle, but it's really through these concerted efforts between many of these different lab groups around the world that are hopefully going to bring an end to malaria in our lifetime. With that, I'm going to pass things on to Aaron.
Okay, good evening, everybody. It's really incredible to see you all here, and thank you so much for coming. And thanks to Jennifer for organizing us and the MIT Museum for having us. Now, this evening I want to talk to you about a topic that's really sort of near and dear to my heart, which is viruses. And I'm part of the Harvard Virology program, which means obviously it's a big part of the topic I study. And I'm a graduate student there working for Pardis Sabeti. So my goals for this are sort of threefold. One is to maybe infect you a little bit with my enthusiasm for this topic. The second is to field any questions you might have heard about things in the news, because sometimes there's conflicting stories about outbreaks or viruses, and maybe you want to get a little bit more feel for what we think is happening. And then hopefully by the end of this, you will know enough on how to prevent the next apocalypse by breaking human-to-human transmission chains. So just to give you a perspective on where I'm coming from, like hopefully some of you, I was actually pretty lukewarm about science in middle school and high school. I enjoyed chemistry club because we got to blow stuff up, and that's always fun. I liked sci-fi a lot, and maybe some of you did too, and I still enjoy sci-fi. And one of the things that really appeals to me about this is the idea that, for example, in Star Trek, you go out into deep space and explore the unknown, and you can see these new alien cultures and learn about what is similar between our cultures and theirs, or how are they different? What is it about their food, their culture, their technology that makes this so just incredible to think about? And in the Sabeti lab where I do work on viruses, I still kind of feel like I'm almost on the same trajectory, not least of all because a lot of us are sci-fi nerds, but also because I would argue that in some ways, viruses are sort of the closest things to aliens that we've ever encountered. Now, there's a couple of reasons for this. The first is that they're sort of just on a totally different scale, both in terms of size and complexity, okay? So let's take a human hair. I've obviously magnified it on the screen for you. It's pretty small, right? Human hair. Human cells, a little bit smaller, and it's pretty crazy to think about. If you look under a microscope, you can even scrape off some of your skin cells, and you can see you are made of billions and billions of these tiny, tiny little critters. And give yourself a pat on the back, because these are really complex macromolecules, essentially, right? They're bags of proteins, lipids, carbohydrates that have assembled, and there's billions of these things going around your body, allowing you to survive and have complex thought, things like that. There's 20,000 genes that allow you to do these complex functions, right? And essentially, of your billions of cells, there's different combinations of these genes that allow you to achieve all these different functions, like heart cells, liver cells, muscle cells, et cetera. Now, when you think about viruses, again, totally different scale, right? I've already blown up this human cell, so it's pretty massive. Can anyone spot the little virus that I've put on here? Raise your hand if you think you can. OK, good, because oh, no, I tricked you, sir. Sorry. I didn't actually put a dot on there because it's actually so small it wouldn't even fit on the screen. So they're on the size of hundreds or thousands of times smaller than human cells. And if you look at their genetic complexity, how many genes did each virus encode? For some extremely successful human viruses, there's only 4. Okay? 4 genes in this tiny particle, this tiny little bit of protein with a little goodie bag of nucleic acids with 4 genes in the middle. And these things replicate to thousands upon millions of copies inside human beings, make the person sometimes sick, and then just go on to the next person, the next person, the next person. All the while, your incredible immune system, which has millions and millions of dedicated cells trying to take out these particles with its 20,000 gene arsenal, is more or less helpless. And one of my favorite virology professors, just paraphrasing, kind of brings up the idea that not only are these so small, but sometimes they're just totally different. You think of every cell, starting from humans, mammals, plants, bacteria, protists, fungi, they all are made up of double-stranded DNA as its genetic core, all cellular life, as far as we know, without exception. Now, some viruses do employ the same strategy, where they have stable genetic DNA, and it replicates at some rate. And in general, you don't want to make too many errors. Now, some viruses, on the other hand, take a totally different approach, and they make their genetic material entirely differently, out of a just different molecule. It's not even DNA. So, like sometimes you say, like, oh, those people are supposed to be identical twins, but their DNA, they don't even look like they share DNA. They're not even made up of DNA sometimes. And that's just, totally alien sometimes. And because of this, some of these viruses have special properties, like they tend to generate more mutations faster than humans, faster than your immune system can adapt. And so they can use their small size, their extremely fast division and replication rates, and then sometimes these high error rates, and that's sort of how they get by. And one of my famous virology professors used to say, every virus that currently exists has defeated every single immune defense that's been thrown at it. If you just think about that, that's kind of like, if this weren't the case, right, the virus wouldn't have replicated and it wouldn't exist. And the little caveat, of course, is it only needs to replicate well enough to be able to spread to a new host. But what really makes viruses different from most cellular life is that they don't do anything when they're outside of cells, right? They're parasites of cells. But outside of a cell, just sitting on the floor, on a doorknob, they're essentially inert chemicals. And so they need some way to spread themselves, and this is why we think of transmission as so important. You can try and treat sick people with antivirals if they exist for a particular drug, or you can try and block the transmission chains. Now, I've put up emojis for animals I could find that I know are infected with viruses, and of course, there's not enough emojis for all the things in the world that are infected, but they really, have the whole spectrum infected. There's viruses that infect animals, bacteria, plants, fish, algae. There's probably viruses that infect every single thing on this planet, and they do so in incredibly different ways. And so you might know, if you have a pet at home, sometimes they can get sick with viruses. So sometimes there's transmission from animal to other animal, whether it be in households, farms, just animals in close proximity. And analogously with humans, right, you can spread diseases between each other. For example, the colds, or seasonal influenza goes around every year. And so there can be direct contact, bodily fluids, aerosols, surfaces, which is why hand washing and things like that are so important, because if it can't spread among people, those viruses will just sit there on the floor, on that doorknob, just as inert chemicals. I'm really fascinated, though, in sort of this interplay between the two, When you get some viruses that seem to be able, for whatever reason, to be able to jump from animals to humans, or vice versa, from humans to animals. And studying some of these viruses is so fascinating because you think about how different humans are from fish or dogs or monkeys or snakes or in some case, for example, bats. Okay, so how many people remember the Ebola virus outbreak in West Africa? Yeah, hopefully not too distant memory. This is just one example I'm bringing up because my advisor and myself were involved in some of this research, trying to understand more about what was happening during the outbreak. And the first thing that made this so different is that Ebola virus was first discovered to infect humans in the 70s, and you can see all these little dots representing outbreaks every decade or so, until 2014, when there was a massive outbreak in West Africa, and that's just giving you for scale, that infected nearly 24,000 individuals, unfortunately, many of which succumbed to disease. Now, there's a lot of investigation onto where does Ebola virus come from, because we only see it once every decade, and you'd expect for a virus like, say, that's really well spread by human to human, like influenza, you see a seasonal influenza, there's outbreaks all the time. But this only shows up once every 10 years, which gave itself to the idea that maybe there's some kind of animal that the virus is hiding in that pops out of. There's been a lot of speculations it's bats. The evidence is still sort of up for debate on that, whether it's really bats or not. But let's say there's some reservoir, okay? We know that it can spread between animals of that same reservoir, like let's say bat to bat in this example. What's also interesting is that if you look at the wildlife around these outbreaks in the past, say the 70s and the 80s and 90s, there's also a lot of infected non-human primates, as well as other animals and wildlife that also come down with Ebola virus disease, which again, they think probably comes from infections from the reservoir into these so-called amplifying hosts. And you can see these massive die-offs of, for example, non-human primates. And then they can, of course, infect themselves. And once they infect their own communities, then there's even more infected hosts, right? And so now we get to the real question. Let's say you have a community in West Africa where this virus has never been seen before, and there's a couple people that are starting to get sick with Ebola virus disease. So these are the people in red, unfortunately. So the question is, how are these people getting sick? There's a couple routes you can think of if this is what we have as our sort of model of what infection looks like. Maybe it's that these people are getting infected directly from the bats. There's some speculation that that's the case. There's some speculation that because there's so many amplifying hosts, the non-human primates, for example, that come into contact with humans, either from, say, like hunting, eating bushmeat, or just sort of living around villages, being in the same area, that there's some infection from that source. Because remember, viruses on the floor, viruses on the doorknob, they just sit there, right? Somehow, there are many infections in the human population. And of course, the last possibility is that it's spreading between people, right? That maybe there's just one early event where maybe somebody got infected from somewhere and then started spreading it among the human community. And the reason this is so important is because different viruses are so different from each other. They have so many different ways of spreading the virus. And all of these are real possibilities that need to be investigated to understand what is the best way to try and prevent a massive-sized outbreak. So what our group and others did was starting to sequence virus isolates from patients infected during the outbreak, and we compared them to the genomes of viruses from previous outbreaks. So here you can see 1976, 95, 2007, 2014. And you can start to build an Ebola virus family tree to ask, okay, let's mix up all these sequences and ask which ones are most genetically similar. And what you find pretty strikingly is that all of these 2014 sequences are like siblings, right? Brothers and sisters, essentially, among each other. They're very genetically similar, for the most part. And when you compare them to the other older sequences from, say, previous outbreaks, they're still related sort of as like cousins or uncles and so on. And you can date this all the way back to sort of a great, because these things replicate very quickly, an ancestral virus. And so what does this tell us? So this tells us actually that if there are just individual infections, say many infections from the reservoir hosts or from the amplifying hosts, you'd actually expect these to not look like brothers and sisters because they would be a lot more genetically diverse. As opposed to these all being sort of descended from one common ancestor, you might expect there to be multiple points. You might expect them to be more like cousins, so sort of more like this relationship. But in fact, because they group so closely together, we strongly have reason to believe that this was all mostly from a small number, maybe one or two introduction events from the reservoir into the human population, and therefore, a lot of the public health officials were able to alert the communities, okay, it's really being spread by predominantly human-to-human transmission. You need to make sure that we do a good job setting up Ebola treatment units, quarantining infected individuals, and making sure that the healthcare workers themselves don't get infected, or else they could spread it to other individuals. Okay. So this is sort of just one example. And again, it's a huge compendium of work that I've sort of just scratched the surface on. Not just our lab, but labs across the world. Truly global effort. And Again, just one example of a virus you may have heard of in the news, but of course, with other ones like SARS, swine flu, Zika virus, I'm happy to field any questions or go into more depth or any questions you might have. So I'll just end. And again, thank you all for being here. Thank you for listening. Hopefully it was somewhat informative. And again, thanks to Jennifer and the museum for having us. Thank you.
Okay, it's very nice to be here. Thank you for coming. So I've been at Harvard as an assistant professor for 6 years now, and I mainly do mathematical modeling. My focus is actually malaria. I'm working with Angela, among other people. But more generally, I'm interested in the dynamics of epidemics, and endemic disease too, for that matter, and how we can intervene, and specifically how transmission works, how different transmission routes work, and therefore how we can contain them. And I'm going to make a distinction here between endemic disease, like malaria, TB, these things that cause deaths all the time, and we're kind of used to it, so we don't have the same fear factor, as opposed to Ebola. Okay, so if you look at the burden of infectious and parasitic diseases, they land squarely on low and middle income countries, especially sub-Saharan Africa and South and Southeast Asia. And in fact, infectious disease still accounts for a quarter of all deaths and 65% of deaths in children under 5 globally. So it's a major burden in terms of morbidity and mortality, and that's kind of an endemic disease burden that we live with every year. So epidemics that I'm going to talk about, and pandemics, which is just epidemics that spread globally, cause us different types of anxiety. And when they threaten the US, we start to pay attention. But in fact, this burden of infection is ongoing. So something that infectious diseases need for transmission is a high density of susceptible people that they can spread between. Okay, so we're living in unprecedented times in terms of the sheer density and size of our global population. So this graph just shows from the year 1000 and projects this way. And you can see we've got this exponential growth in our population, which continues unabated, and particularly in Asia. So this is from the Washington Post, and in this circle, there are more people living in this circle than outside of this circle. So a lot of my work is now focused on trying to understand how we can prepare for and project and control epidemics occurring in South and Southeast Asia in particular. And one of the reasons it's such an important place is because we have massive urbanization, huge rural to urban migration, and incredibly high density of populations in urban areas, peri-urban areas. And so that's perfect fuel for epidemics to spread. They need susceptible people. This is an aerial shot of Manila, which is one of the most densely populated cities in the world. And you can imagine if you were a virus or some other kind of pathogen, this would be a pretty good opportunity to spread very quickly. So another thing that's happened, in addition to population growth and increasing population densities, is that we are incredibly connected these days as a global population. So this is these maps show the lifetime extent of travel of 4 generations of men in the same family. So the author of this paper, this is his travel trajectory, which is going to be familiar to many of us. I think I did this last week. This is his father, his grandfather, and his great-grandfather, who in his lifetime traveled no more than a couple of tens of kilometers. Okay, so if you are an infectious disease pathogen and you want to spread, this is the way to do it, right? If you have local transmission occurring, spatially contained, it takes a long time. So even the Black Death, enormous mortality, huge impact on the world. But it spread spatially quite slowly because populations were less dense and people didn't travel that far. So we're now living in a world where you can't think about national borders as a sensible boundary for control programs. We live in a global world, and we're not safe for epidemics. And simulations are pretty clear that closing borders is not a very sustainable or sensible solution for many infections. So this is a map showing the spread of H1N1 and that you're all probably familiar with. Global travel spreads infection very rapidly. So I mainly do mathematical modeling, and we use dynamical models. And to illustrate why we need them, why we can't simply gather data and make statistical models, so just to kind of describe the data and make a projection, I think it's useful to think about why it's different from a non-communicable disease, right? We talk about the epidemic of diabetes or cancers. And for non-communicable diseases, you can think about the population outcome meaningfully as the average of many individual outcomes, right? So it's like lots of people playing pinball. We have complexities and probabilistic dynamics happening for each person, and we're all playing separate games, and then we can average the outcomes, right? Because they don't depend on each other. I mean, there's some issues with whether social networks are a means of contagion of obesity and these kinds of things. But for general purposes, that's true. For communicable diseases, my outcome depends on the outcome of my social contacts, right? And this is why the vaccination debate is so important, because whether you vaccinate your kids matters for somebody else's kids. And that's because the individual risks and benefits can only be studied on a dynamical level, thinking about everybody's outcomes. Because the more disease there is in the population, the more at risk you are. So we need dynamical models that can incorporate feedback loops and relationships between people. So I'm going to briefly talk you through what epidemiological modelers do when they think about modeling epidemics or making projections. I'm sure a lot of you saw the projections made about Ebola. How many people will die? How many people are going to get Ebola? Policymakers want to know these numbers for obvious reasons. The models are surprisingly simple, and I'll sort of describe them. I won't go into the maths in much detail, but essentially we understand a population as a bunch of susceptible people. There's an infectious person comes into the population, right? So we literally classify our population into groups in relation to their infection status, right? So everybody starts out susceptible if it's something like Ebola in Conakry. An infectious individual enters the population and then spreads the infection through the population, okay? So you can be susceptible or you can be infectious, and then eventually you might recover, right? So if it's measles, you become immune to measles, you can't catch measles again. Okay, now, so this is a simple model. It has 3 categories of people, and you model the dynamics whereby your rate of flow between these categories of individuals depends on the density of infectious people and the density of susceptible people. It's fairly simple, and it yields dynamics like this. So this is a simple so-called SIR, susceptible, infectious, recovered model output. You have everybody susceptible at the beginning. The infection slowly spreads and sucks up these susceptible people. And then the epidemic runs out of fuel, runs out of susceptible people, and it peaks and comes down, and then our recovered people come up. Okay, so it's literally a model as simple as this that was first used to understand Ebola and make projections about how big it was going to be. And yeah, so this simple kind of workhorse that we use is surprisingly effective. So epidemics do kind of look like that, right? This is an outbreak of dengue in Pakistan in 2011. Dengue is a mosquito-borne virus. It's transmitted by the same mosquito as Zika. So these populations are all susceptible to Zika, although there may be cross-reactivity, which would be interesting. So it's actually a useful model because it accurately describes how infections spread through a population. So we can do a good job of predicting dynamics of epidemics if we have a sort of a closed population and we know what's going on. But for Ebola, and this is data from Guinea, actually, and it shows that these little transmission chains that need to be initiated to start an epidemic off were coming in from outside, right? So first introduction, I love this, uncooperative family. They didn't do what they were told. There's more transmission. And again, these introduction events happen because people were moving in from outside, right? So yes, we can model a closed population where we have mostly susceptible people and an infection comes in and it takes off. But we need to know how populations are connected if we're going to understand transmission more regionally and try and intervene on transmission generally. So how we do that is we take our simple SIR models, and then we divide up our population into subpopulations that we think define these little clusters of people where transmission occurs within them, and then there's migration between them, right? So we build up this picture of clusters of people, and this could be cities, it could be villages, it could be countries, and all of these models were made for Ebola. And so you link your population. So we say, okay, we know that there's high risk over here, there's moderate risk here. How many people travel between these places? How likely are they to be infected? And similarly, if you're a resident, how many people move to high-risk areas and come back and they could bring the infection too? All right, so we need to know this if we're going to move from a simple single population model up to a regional spatial scale that's useful for containment questions. So when we think about this, the first question is, okay, well, we need data. We need to know how people are moving around if we want to contain epidemics. Okay, so I like this graph. It shows the impact of spatial and temporal travel patterns on the spread of infections. So here on the x-axis, we have the spatial dimensions of travel, going from neighborhood travel, like going to the store or going to school, all the way up to international. And on the y-axis, we have daily travel patterns, commuting and that kind of thing, all the way up to long-term migration. Okay, so local movement patterns are the kind of underlying fuel for transmission in a closed population. And you can think of this as your kind of SIR model that you would do for a single population. But then on a regional scale, and particularly if you think about domestic containment strategies for epidemics, you need to know about this. How are people moving around the country? How are they moving between cities? And then, of course, there's global spread, and there was a lot of, there were travel restrictions put in place, and airline networks were analyzed to look at that. The problem is that we don't have good data right in the middle here, or we didn't until recently. So the local travel, there's been GPS studies, there are travel surveys. Travel surveys are terrible because no one remembers anything about where they went, ever. Census data is good, but it happens on a yearly timescale as things, where did you live last year, right? That's not really on the right temporal scale for outbreaks. We can use airline and shipping networks, and those have been very effective for thinking about international containment. But there's this big gap here where we don't have good information, especially in low and middle income settings. Okay, so something that we're doing in my group and has sort of gained some traction is that we are looking for other ways to get data about population dynamics underlying epidemic spread. So this shows the increase in mobile phone ownership. And I'm sure you've seen this type of graph before. It's the rapid adoption of this mobile technologies. And it's not just in high income settings, but it's also increasingly in low and middle income settings. Penetration of mobile phones is amazingly almost complete. I think we have a study in Thailand, and the number of SIM cards per person is like 150% or something like this. So what can you do with mobile phones? So operators store data whenever you text or call. And these are time-stamped, and they are associated with a cell tower location. Okay, so you're making calls, and it's logged by the operator that you're calling from tower A, okay? And then you move over to tower B, and your time-stamped calls are in the log, and then you move to tower C. So using some maths, you can piece these together into a longitudinal picture of where that person went over time. Operators use this data for churn analytics and other reasons all the time. It's free, it's in real time, and it's scalable in a way that travel surveys and small GPS studies simply aren't. If you work with operators to anonymize this data and aggregate it effectively, because subscriber privacy is obviously a big deal here, and we can discuss that if you would like. But what it means is that at the scale of entire populations, millions of people, we have information about where people are, volumes of travel between location A and B, between cities. Tower density in cities is so high that we can get location down to, half a kilometer by half a kilometer. So this is extremely powerful information. We know where people are, and we know where they're going. So we have been working to use this information to try and model transmission of infectious diseases in space, with the question being, where should we place resources? how can we prevent spatial transmission of infection in a sustainable way. So this is the first study we did was in Kenya, and I'm a malaria researcher, so we started with malaria, which is very spatially patchy in Kenya. And so what we did is we looked at mobile phone data from millions of people, and we modeled where people mainly traveled from and to. Okay, so if you look at sources and sinks of travel, so you basically ask the question, where are most people coming from and where are they mostly going to? That's this picture here. And this is Nairobi, which is the capital. Lots of people travel to Nairobi. It's pretty intuitive. And then there's a lot of heterogeneous travel all around these areas that correspond to highly populated regions, basically. Okay, so then what we do is we combine these with our SIR kind of models, right? So we have our SIR models within regions, and then we use the mobile phone data to parameterize migration events with probabilities that people are infected. Keep in mind, we don't know which people in the cell phone data are infected. This is a mathematical model exercise. But what comes out of that exercise is that the source of malaria for many of the cases in the rest of Kenya is coming from this one region. A lot of it's going to Nairobi and these highland regions. And what that does is it changes your intervention strategy from we need bed nets everywhere, let's just measure where malaria is, to why don't we target the source of infections rather than continually mopping up imported ones? Okay, so that's the kind of general idea, and we've been conducting studies in different places. We did a study in Pakistan looking at the transmission of dengue between cities. So this is Lahore, and you can see the black dots are the location of cell towers. It's very high density of cell towers in cities, and that means we have very good high resolution picture of where people are and where they're going. So we can map them in relation to hospitals, those are these green places, and figure out things like how mobile are populations near the hospitals? Do we think they can have access to those hospitals? What's the catchment area for surveillance and what should it be? These kinds of questions. And what we found was that there was this enormous volume of travel. This is a very large distance. I don't know how many of you have been to Pakistan, but this is very far away. And our standard models that we would have used before simply would never have predicted that sheer volume of travel. And so when we put this into our dynamical model framework, essentially an SIR model, but with some vector components and environmental inputs, we can do a good job of predicting where dengue outbreaks are going to happen, significantly better than previously. And in particular, we predicted outbreaks in places that wouldn't have been predicted otherwise that were correct. So it seems to be a powerful approach. And then, of course, discussing Ebola, one of the reasons Ebola took off so dramatically in 2014 is because prior to that, it had only occurred in rural areas. So, I was talking about the risks posed by the growth of these cities and slum areas in particular. For Ebola, it emerged in a rural area, but then in this highly densely populated, connected area of West Africa, it got into specifically city regions and peri-urban slum areas, and that's when it took off, because you had a high population density of susceptible people, frequent contact, and a highly mobile population. And so we developed some of these maps based on some limited mobile phone data to try and help project and understand where to place resources. There were many mobile health clinics and this kind of thing. Okay, so I guess I'm just going to end by saying that these pandemics are a feature of the 21st century and will continue to be so. Because that's the way our world is now. We have increasing population densities, rapid population growth still, and incredibly high connectivity, incredibly high mobility. And so we're starting to think about ways to prepare for these pandemics. And I think pandemic preparedness poses all kinds of financial, economic, political challenges. We have to cooperate regionally and internationally because infectious diseases do not respect borders. And in particular, we have to be preparative as opposed to reactive. Every time there's a new epidemic, we all jump on it and we try and get funding and we try and research it, and then it goes away and there's the next one and we jump on that one and we try and develop a vaccine really quickly, and that's important. But preparedness is something that's going to be critical to avoid massive massive outbreaks in the future. So I hope that's not too depressing note to end on. But thank you very much for having me, and I'm happy to take any questions. Oh, not yet. No, I'm not.
Fantastic. So I think you guys have a lot to mull and chew over a little bit. So now comes the discussion part of the evening. So you're sitting at tables. Hopefully you've had a chance to make some new friends. And so now you're able to sit, discuss, and talk about what you've heard, what questions you still have, and what your thoughts are. We'll be passing around some iPads, so you can also tweet in your own questions. One thing, though, you must include hashtag MIT Soapbox, or we won't see your tweet. You can also just raise a hand and ask a question when we get to the Q&A portion, but I know sometimes this is an easy way to ask a question if you aren't always certain about asking questions. But yes, you have about 20 minutes, so sit, talk to each other, grab a fresh cup of tea, and have a good conversation. So let's move on to our Q&A portion of the evening. So our speakers have been reading this Twitter fall as well. If you guys have questions, you can raise your hand. I'll be coming around with the microphone so we can hear you. And then they'll do their best to answer and respond as well. So why don't we actually start with Carolyn? You said there's something up here that was speaking to you.
Okay. Yeah, I'll go first because I have to leave because small children need to go to bed. Okay, so I will respond to this. Do you think the public has an accurate grasp of the threat of infectious disease? We humans are notoriously bad at understanding low probability events that have very high risk. So we routinely overestimate the risk of terrorism, for example, and other things like, I believe, pandemics, we underestimate or we just don't think about the risk. So we're really bad at this, right? We like get scared of flying, but we're happy to get in an Uber with a random person and drive across town. So no, I don't think they do, but I think this is part of being human, right? Humans don't understand low probability events that have high hazard. I can let you do the virus one. Okay, what can you do to reduce the spread of infectious diseases? Don't have children. Yeah, I think being aware, right? Like, so I travel a lot, and even though I'm traveling to think about infectious diseases, very often I'm like, I wonder what infections are in that country that I'm about to go to. I'm probably vaccinated, so that's good because I'm vaccinated against everything. But I think being aware, right, go to the CDC and think about what infections, if you're going somewhere, think about it, get vaccines. I'm sure I'm preaching to the choir, but vaccinate your kids. And then I think there are also, specific times of year that pose high risks for infections. And to be honest, a lot of the prevention things that we have are still pretty basic. It's hand washing and some of these things. Not shaking hands. Shaking hands is a pretty bad one for spreading infections. But again, I'm not a clinician. And then I can say about the malaria vaccine, and Angela can elaborate. So there is a malaria vaccine. It's been, there are clinical trials, major clinical trials that have happened. Angela talked a bit about the complex life cycle of the malaria parasite. One of the things about it is that it has a pre-blood stage, right, in the liver, and then it goes into the blood, and it's only when it's in the blood that it causes any symptoms. And the vaccine, so the part of the parasite that you're generating immunity to, the protein in the vaccine that is actually a pre-blood stage protein. So what that means is it's not attacking the part of the parasite that is actually causing symptoms in the blood. So one of the things about malaria is it's enormously genetically diverse. There are many, 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 many strains of malaria. And so what that means is when you're trying to develop a vaccine, you have to decide which strains to put into your vaccine, just like we decide for the flu vaccine every year which strain to put in. And sometimes they get it wrong, and sometimes it works. But for malaria, that problem is magnified many-fold. There are so many thousands of different variants out there, and the vaccine that's currently being tested is just one type of protein. It does seem to provide protection against some severe disease episodes in small kids, and it puts off your next infection. So it's doing something that's causing partial protection. Whether or not it's the solution to blocking transmission, I think is much less certain, right? And this is a talk about transmission. The vaccine that's currently being trialed and will probably go out is not a transmission blocking solution, but it does have some efficacy against severe disease, it seems like. So I'm sure you can elaborate on, I'm gonna make Angela talk about gene drive, but that's very cool. So anyway, thank you very much for having me.
Thanks for coming. Do we have any questions from the audience before we continue into some of the Twitter questions? It's okay if not, just wanting to give everyone equal opportunities to ask questions. So let's move into the gene drive. If anybody had been here during last year's soapbox on synthetic biology, we also started covering gene drive. So it's really great seeing these topics bridging between our other topics.
So gene drive, this is a bit of a contentious question. For those of you who aren't familiar with the term, gene drive refers to, it would be creating a form of a genetically modified organism, usually using this technology called CRISPR, which some of you may have seen in the news. It's been sort of a hot topic in genetics these days, although there are other gene drive systems. And essentially what you do is you put a gene into an organism's genome that is going to sort of artificially propagate itself in some way. So in other words, during sexual reproduction, people may remember you get one copy from the mother, one copy from the father for each gene. In a gene drive system, you're always going to be more likely to get a certain copy from your mother or father, depending on what that gene is. So essentially, at the level of just making these gametes, at the very basic level of reproducing a new organism, this special gene drive system is going to have a way of sort of forcing itself to the front of the line in each of these reproductions. So with the exact question, how it could be used for something like vector systems, one of the common things that's talked about, and remember, none of this is out there on the market yet. These are all just things happening in the laboratory. There's a lot of ethical discussions that need to go in, legal discussions that need to go in before anything like this actually is released in the wild. But one of the main things that's being investigated is that type of sterile male mosquito technique that I mentioned a little bit in my talk. So the idea being you could put this gene into mosquitoes that actually makes the males sterile. And then when you release that mosquito into the world, it may actually still be able to reproduce in some way, maybe through females or something like that, but then there's going to be some sort of dead end that would happen. So that would be sort of one example. Another example, which would be harder to do, would be to maybe just change the malaria parasite, the mosquito in some way that it doesn't let the malaria parasite in. So maybe it'll take up a blood meal and that mosquito will have that malaria parasite in its gut, but that parasite now needs to develop for 2 weeks and find a way to reach the salivary glands of the mosquitoes. And it's only able to do that in very specific malaria transmitting mosquitoes. So if you had a gene drive system, very hypothetically for instance, that could change the receptors, the form of those mosquito salivary glands, then maybe that mosquito would just start blocking the Plasmodium parasite and it wouldn't be able to enter it at all. Of course, there's, evolutionary selection, lots of questions that still need to be answered, but those are sort of the what-ifs that people are looking into now.
I feel like that could tie in really well with this other question about the role mosquitoes play in our ecosystem. And so if perhaps gene drives were introduced to reduce the mosquito population,
Yep.
What type of impacts could that have?
No, for sure. Like if you're talking about reducing mosquito populations, this should be something that immediately pops into people's mind. A few things to keep in mind here would be the fact that as much as possible, the goal is if you're trying to eliminate or reduce mosquito populations, the goal is to do it in some sort of targeted way. And gene drive would actually allow you to do that, which is one reason people like it. So instead of just killing all mosquitoes in the ecosystem, you'd just be targeting those handful of species that would actually be capable of transmitting diseases. So instead of targeting all 3,500 mosquito species, you target the 30 that could actually transmit malaria. The idea being that then those other mosquitoes are still out there in the ecosystem kind of carrying on these roles. In terms of what those roles would be, one main role is the fact that mosquitoes actually have a larval stage that's in the water, if anyone's ever heard of wrigglers or seen them. And this is why you're always told to dump any standing water that you have outside, especially in areas now like in the south where there's Zika. Because mosquitoes are able to breed in just a tiny little few teaspoonful of water, and that's because they have this larval stage. And those larvae actually do play a role in aquatic ecosystems. They're eaten by larger insects in there and can reproduce through that. But in all honesty, most mosquito research goes into those 30 or 40 mosquitoes that are actually capable of transmitting diseases. So I know there's a lot to still know and learn about the more benign species that are out there.
Aaron, do you have anything you want to comment on that's been showing up in the Twitter feed? Or anything that you heard that you want to comment on?
Sadly, I don't know that much about malaria myself, but definitely a lot of my colleagues, work in sort of similar fields of infectious disease, and a lot of these challenges are really similar. I'll kind of take this 1 second from the top, because I find this really fascinating myself. The question is, didn't we all start off as viruses? Mitochondria. Now, this is like a serious contention of debate within the scientific field, and one of the really fascinating sort of theories that's emerged is, okay, so I told you that some viruses, so all cellular life uses double-stranded DNA. Some viruses also use double-stranded DNA as their genetic material, but some actually use RNA, which is, like I said, a cousin to DNA. It makes up, it's sort of the more direct messenger that gets translated into proteins, and it has all these functions that by itself can do some kind of like cool enzyme stuff, so they actually can do some things. Whereas we think of DNA more as like a template, they're building blocks, and maybe they've got some functions, but they tend to kind of be more of like your stable building blocks rather than the actual enzymes that kind of go around doing things. And so one hypothesis that's been put forward is that we've got 3 essential major kingdoms of life. There are eukaryotic cells that include all of mammals, plants, and then there's essentially one for bacteria that we think of like E. coli that you might have heard of, and then one for sort of like older E. coli, older bacteria that's a little bit more distantly related. And what's been proposed is that maybe there was a 4th branch of life a long time ago, one that was essentially an ancestor to modern viruses, okay? So the thinking is there's some kind of something out there, right, way back in the primordial soup, and at some point, for whatever reason, it like lost the ability to do what cells do, which is grow on their own outside of cells. It lost the ability to utilize energy and translational machinery and make its own proteins. And so that's how somehow it just became these inert viruses that just sit there outside of cells. And one line of evidence to support this has been the relatively recent discovery of these giant viruses. So if you remember, I tried to trick you into looking for a little dot on that speck, but there's some viruses that have been discovered in the past couple years that are gigantic. And that's still pretty relative, right? So like, most viruses kind of have between 4 and 100 genes. And so when I say, and again, human cells have 20,000. So when I say gigantic for a virus, that means, these things have a few thousand genes, okay? And they're actually, some of them are so big you can see them in a light microscope. So they're still smaller than human cells, but they would actually show up as like a dot, like a not insignificant dot on that slide. And what's fascinating about these is these are almost like hybrids, like in between real cells, because they've got some of the machinery to make proteins, but not all of it. But you could imagine almost if they just lost a few genes or just gained a few genes, they could actually be kind of a functioning cell. They're not that different. So some of these questions are really emerging now when we have the ability to do sort of genetics and genomics, and that's why I'm so excited to be working at the Broad and cool places like Cambridge, where a lot of this work gets done.
Fantastic. Do we have any? Yes.
Yeah.
I've always wondered how do you really build phylogenetic tree for viruses,
Yeah.
given how high mutable they are. But also, I just want to make a comment on the question, because I believe mitochondria is bacterial origin and it actually have DNA material. It's not exactly virus.
It is or is not?
It's not. So mitochondria is supposedly from
Yep.
DNA material for bacteria,
Yep.
and it's symbiotic to human is not exactly viral per se.
Yeah, okay. So I'll take the first part of that first.
And will you repeat the question?
Yeah. So the question was, I'm blanking.
How do you build
So in two parts.
connection for
Right. So first part of the question, how do you build a tree, a family tree, sort of like Ebola for some viruses? Now, thankfully, during, for example, Ebola virus outbreak, it's actually a fairly conserved virus. So if you sort of look across the genomes, there's actually not that many differences compared to a lot of other viruses. Some viruses, like influenza, are so diverse from each other, it's actually really hard to do what they did here. One strategy to sort of get around this is, instead of looking at the genetic material, you can look at the proteins that these viruses encode. And if you look at the actual proteins they encode, they tend to be more similar than the actual genetic material themselves. And so that's one way to get around it. But if you wanted to make a tree of, for example, every single virus family, certainly people have tried, and it's really difficult. I think there have been some really clever approaches, again, trying to look at things like proteins that tend to be more similar compared to genetic material, and look at protein structures, which we think are even more conserved than the protein sequences, which are more conserved than the genetic material. But those are certainly really, really tough bioinformatic challenges. The second part was about mitochondria. So the current sort of dogma in the field is that bacterial cells, kind of these globs of materials, they don't have any sort of like guts inside them that we think of for eukaryotic cells or human cells or other types of cells. And in particular, our human eukaryotic cells have these little organelles called mitochondria that are powerhouses of cells. And what's remarkable is they look a lot like little bacteria, as was mentioned in the question. And so at least in that sort of case, it looks like what might have happened a long time ago is that a kind of biggish cell swallowed maybe a smaller bacterial cell and sort of used that as its energy powerhouse. And there was eventually a symbiosis. I think the role of viruses in sort of ancient mammalian genes is really only starting to become recently known. So for example, if you look at the human genome, just in the nucleus, so not the mitochondria, so like you think of your normal human genome, about 5% of that DNA, maybe more, maybe up to 10 or 15%, comes from, we think, events where an ancient, say, retrovirus, so similar to HIV, but obviously not HIV, so an ancient virus that's related to HIV gets into non-human primate genome and integrates, and then just kind of sits there. And over time, these events kind of keep accumulating until you have 5% of most human genomes nowadays have these signatures of retroviruses that long time ago seem to have integrated into human genomes, for example. People have been looking at that for mitochondria, which is a similar story. And I'm not totally up to date on that literature, so I don't have a great answer for you there, but certainly people are trying. I think there's some interesting work to be done.
Fantastic. Thank you. So that brings the end of our evening up. But I hope you guys all had a great time here, and hopefully you'll consider joining us for future programming. Let's give our remaining two speakers a round of applause.